Good afternoon, everyone. What I'm going to do in the next 15 minutes is just give you a highlight of who we are, who Precigen is, and what do we do, and how we consider ourselves differentiated in certain platforms. So with that in mind, obviously some forward-looking statements here, but basically what we have, and we are somewhat unique, is we have two platforms, both cell and gene therapy. Now, we have the gorilla adenovirus platform for gene therapy, and then we have our Ultra CAR T platform for cell therapy. And the way we look at this and why the two of them together is because when you're looking in immuno-oncology, your reality of the immune system is there is not one fit. There is not one silver bullet. What you have to do is sometimes you have to look at your immune system as a general army that the body has. Now, that army, it can be trained sometimes in vivo directly with certain training or forces like our gorilla adenoviruses, vectors that they vaccinate and they can generate high affinity T cells. On the other hand, there is a part of the immune system that requires basically taking it out and specializing it further to see a specific of the enemies from within. And that's the UltraCAR, and this is how we are bringing these two together. And no one says that actually these two things cannot be combined. They can and they should in the future, and we are going in that direction. So our gorilla adenovirus, our gene therapy, which I'm going to spend some time first, 5 or 10 minutes on that, is differentiated from other AAVs. In what way? First of all, we have very little pre-immunity, almost zero, to these vectors. What does that mean? You can keep repeat dosing them. When you repeat dosing, what we have seen, the patients, they basically, the T cell responses keeps going up, whereas the neutralizing antibodies do not. And this is very unique only to these vectors. You do not see it with other AAV. They have a high capacity. We can go up to 12 kb of various epitopes and genes, which is unlike the others. If you're looking at AAVs, 5, lentiviruses, retroviruses, all of that. And also we have a very highly productive manufacturing process for this. On the other side, for UltraCAR, what we have done is, before we even started conceptualizing our clinical, we wanted to differentiate. There was no sense for a biotech company like us to compete with Novartis and Kite in the same space with the same type of a CAR T. So we went and looked at what are the situations that we have. How should we differentiate? The classical CAR Ts, or TCRs, or allogeneic. One thing that they all have, first of all, use of lentiviruses. Expensive, long manufacturing, because you have to get the homogeneity. Then you have to infuse hundreds of millions, if not billions, of T cells that they only survive a few weeks. And why they survive a few weeks? They are exhausted as you enter them. And why they don't work in a solid tumor is very simple. Everyone says they don't traffic. It's not that they don't traffic. They just don't have time to get to the sites that they need to go in a solid tumor. Versus when you are in a hematological setting, of course they see the antigen immediately and they can react, and they have 2 to 3 weeks to clean up. And that's exactly what happens. So in order to have, for solid tumors also, efficacy, what you need is CAR Ts that are not exhausted. They can get to the sites. They can maintain and persist in the absence of an antigen while they are getting to different sites. And then you can control them to activate them and expand them. And those are some of the major differentiation. One other things that we wanted to get away from a lentivirus was to use a non-viral, and I'm sure the whole audience knows non-viral platform, electroporation, has been really challenging in research as well as in development because of, first of all, the transfection efficiency and the issues that are associated with the viability. So we had to overcome that in order to go to the non-viral, and when I get to the UltraCAR, I will explain how we did that. So with that in mind, our strategy is really to generate value over the period of time based on the two platforms that we have. And how we have done that, just to give you a snapshot of this, with our adenovirus platform, we have gone to rare disease as well as to oncology. And currently, we are in the process of finishing our clinical data, and we have received the both breaking through as well as accelerated path for approval from FDA in a rare disease. And we are going to submit for second half of this year, preparation for a BLA. On the other hand, we also have used it for our another product, Precigen 2009, which really directs toward HPV-infected or associated tumors, such as cervical cancer, head and neck, and that's in phase 2. And our next generation of cell and gene therapies is affecting our UltraCAR-T. So this is our portfolio of the clinical trial. On the top, you have the adenovirus platform, and in the bottom, you have all of our UltraCAR-T platform already in the clinic, minus CD19 that is moving toward the clinic very fast. Let me tell you a little bit about Precigen 2012, and this is from our gorilla adenovirus. First of all, this is a rare disease that you get this because of the HPV 6 and 11 infection, and this can happen in childhood or in adults. You get these benign tumors that are recurrent continuously on a vocal cord as well as in the trachea. The patients cannot speak, they cannot breathe, and there is nothing for this patient except surgery after surgery. These one of the areas of the vocal cords and trachea, those are the tumors. And what you have here is basically that the growth of this, it's like a grass. It grows, the surgeons, they cut it, grow back, and it just gets worse over a period of time. Some of the patients that we have seen, they have had between 200 to 300 surgeries, averaging somewhere between 6 to 10 surgeries per year. So what we have done is using our gorilla adenovector, which keep enhancing the T-cell responses without increasing or generating very little neutralizing antibody. We have developed a vaccine that you give in a span of 85 days. We enroll the most severe patient population. It's defined as at least minimum of 3 surgeries per year. And it's given subcutaneously to these patients. It generates a very strong T-cell immunity to HPV 6 and 11 to basically go to the root of the problem as opposed to just cutting the grass on top. So this is how it's done, and the trial endpoint is we have been following the patient as a complete responders that they don't require any surgery for 12 months, and obviously we have been following them post that. What you see here is that out of the 12 patients that they were basically dosed with the dose level 2, 50% of these patients, they did not require any surgery after 12 months. Actually, these patients have been continued to be in a complete response. They have passed the 24 months, and some of them are in 3 years response rate. For a disease, and these patients on the yellow bars, you could see the number of the surgery that they had prior year, and then on the right-hand side, you see what it happened. Overall, if you look at all of the patients, there was 83% response rate in reduction. And this is with only giving one course of this vaccination at this point. We also published extensively on the science of this in a science translation and show a very good correlation between the response rate and T-cell immunity that has been generated for a long period of time. More importantly, the safety of this. You give a sub-Q, and this is the safety is basically like a flu vaccine. This is how our investigators have said, and patients. There is no grade 3, grade 1. There's slight rash, a little bit of fever in some patients, and the next day it's gone. So based on that, FDA granted us last year our single-arm phase 1 and single-arm phase 2, actually as a pivotal, a breakthrough designation, and moving toward the accelerated path, which we are very excited about for our patients and for our organization. And this is something that are basically some of the KOL and investigators in this field say about this drug product. The safety of it, the ease of administration of it, the efficacy and the durability that you see for a long period of time. We are poised to actually present the phase 2, complete phase 2 data by the end of this quarter. So, and in the second half of this year, submit our rolling BLA. So let me tell you a little bit about our UltraCAR. How our UltraCAR is the lower corner. We use a plasmids that they code for your CAR of interest, membrane-bound IL-15, and the safety switch. This is the essential part of every CAR that we put in the clinic. And why that? First of all, CAR is the CAR, has to recognize the cancer cells. Membrane-bound IL-15 only signaling to the CAR, not across, because then you're gonna make the CRS even worse. And the safety switch. If anything goes wrong, you can eliminate these cells. And they're all under the same promoter, therefore, you have the homogeneity of the expression. We also put our own ultraporator. This is a semi-closed electroporator machine that we developed as part of our manufacturing device. You can electroporate 4 billion cells under basically 12 minutes, so you can scale up. And this is an overnight process, end-to-end, at the hospital. There is no centralized manufacturing. There is no activation of these cells outside. There is no expansion, and it's autologous directly to the patient. This is the difference in differentiation between a CAR, UltraCAR, versus a classical CAR. It's basically happening in hospital, and truly vein to vein is overnight. So with that, I'm going to just very quickly just show the highlight of one of our trials. We presented at ASH last year, the phase 1 data. This is in completely refractory relapse AML patients that they have 2 to 3 months to live. Some of them, they came out of a hospice to receive this. Why this platform is important is because you can produce these cells overnight. They are not activated, therefore they are more stem-like. They manufacture themselves in the body of the patient. They expand and persist. You can see on that graph, basically, these cells, as they enter, they expand when they see the antigen of interest, in this case CD33, and they come and they persist. We have had patients that we followed for 270 days, and you see these cells very well. What we have shown at ASH, we had close to 30% objective responses. CR, PRs, stable diseases. And in this patient population, that is really very, very encouraging. And on the right-hand side, you see one of the case studies. These are extramedullary AML patients, which are different because they almost have like a metastasis looking. What you see in the majority of those dots in the black, those are tumors. And what you see on the right-hand side, after receiving 28 million cells, these patients were completely clean, except that one spot on the head. One thing about this platform, the maximum doses that we have given, it's around 70. It's 1 million cells per kilogram. And you can see responses as low as 7 million cells per patient. So with that, we also are forwarding our CD19, the next generation, because in this one, not only you have the 3-gene CD19, but also now intrinsically we are lowering the PD-1. Why is that important? I'm sure you heard from a lot of other talks. Everyone, CAR T or no CAR T, you have to combine with the checkpoint inhibitor when you send this army to the tumor microenvironment. That is systemic toxicity plus addition of the cost, another $150,000 that you have to add, almost $500,000. Here, only the CAR T reduces its own PD-1. This is moving very rapidly toward phase 1. It will be quite exciting, especially for autoimmunity, because this is very low cost to produce, it's fast, and you have safety switch. If anything goes wrong, you can eliminate these cells. Whereas FDA might be very tolerant at the oncology indications of stage 4, on a chronic diseases will be a different standard. So that's the newest. And finally, this is our vision, and I apologize, I just ran out of my time. This is the vision for precision medicine. The way we look at our Ultra CAR T is, and currently, by the way, this is happening at Mayo Clinic, Moffitt, Fred Hutch, University of Washington, not by Precigen personnel, but with the clean room, the same room that prepares the bone marrow. They do this. The patient comes in, they get their tumor molecularly profiled. The oncologist will ask the clean room, basically, prepare an CD33 CAR, prepare a mesothelioma CAR. The next day, the CAR is prepared because everything is overnight at the hospital. There is no centralized manufacturing. And within the first few hours, the next day after harvest and characterization and a release assays, it will be infused. Fresh product is infused back to the patient the next day. And with this, we believe this changes the paradigm. You don't have the cost of manufacturing. You don't have the time. We heard yesterday what the families have to go through even when the CAR Ts are being prepared. End of the day, you can redose as many times with a very low cost because this is a plasmid and the electroporator at the side of the hospital. And finally, if there is a mutational escapee, which in the hematological side, there is a lot of them with the indication, you can dose with a different CAR. You can mix CARs together. And you can do that. This is why we call this a library approach. So with that, I thank you for your attention, and hopefully I have given you a sort of review of what we do at Precigen. Thank you.